Thank you, everyone, and welcome to our talk on labeling for biological products. As Jeff just mentioned, I'll be tag teaming with Ruby. First, we have the standard disclaimer slide, which I'm not going to bore you all with reading, but it's there. And to quickly go over our agenda, we'll be providing some high-level background about biologics and biosimilars, and then we'll be addressing two substantive labeling-related topics. The first is labeling in biosimilars, and the second is labeling in transition products. I'll get into a little more detail about what we mean by transition products later in the presentation, but as a quick overview, a transition product by transition product, we're referring to biological products that were approved under Section 505 of the FD&C Act that on March 23rd, 2020 will be deemed licensed under Section 351 of the Public Health Service Act. So just to quickly delve into that background, the first slide we're going to discuss is basically what is a biological product. A biologic is produced in a living system, such as a microorganism, like a yeast or a bacteria, or plant or animal cells. Most biologics are complex molecules or mixtures of molecules. In contrast, what we call small molecule drugs are typically small single version active ingredient that results from combining specific chemicals in a ordered specific process. And the picture on this slide is intended to visually demonstrate the difference between a biologic and a small molecule drug. You can see in this picture that aspirin has 21 atoms compared to a monoclonal antibody that has about 25,000 atoms. And the monoclonal antibody also weighs about 100 or 800 more times than aspirin. With respect to biological product regulation, there are currently two different pathways for BLAs. The first pathway is known as the 351A BLA, or a standalone BLA, and it contains all information and data necessary to independently demonstrate that the proposed biological product is safe, pure, and potent. And for a very long time, that was the only licensure pathway under the Public Health Service Act. In 2009, the Biologics Price Competition and Innovation Act was enacted, and it created an abbreviated licensure pathway known as the 351K pathway for biological products that are shown to be biosimilar or interchangeable with an FDA licensed reference product. And by statute, a reference product is a biological product that's licensed under Section 351A, and it's the product against which the 351K product is evaluated. Biosimilars and interchangeables are subject to FDA's rigorous approval standards. One thing to note is that generally their development does not require as many expensive and lengthy clinical trials as a 351A BLA. In addition, the BPCI Act also amended the definition of biological product to include a protein except for any chemically synthesized polypeptide. And I can get into more a little bit later, but why this is relevant is whether something meets the definition of a biological product is relevant to whether the product will be subject to a BLA or an application under Section 505 of the FD&C Act. And it also relates to whether or not a product will be deemed to be licensed under Section 351 of the Public Health Service Act.
Good morning, everyone. So for the next few slides, I'm going to focus on labeling for biosimilar products. So once again, these are applications submitted under the 351K pathway, abbreviated licensure pathway. The labeling for biosimilar products, this particular guidance was finalized July of 2018, and this guidance provides detailed recommendations on developing labeling for biosimilar products, specifically the prescribing information and patient labeling. FDA will provide additional recommendations on labeling for interchangeable products, so do be on the lookout for that. Generally, a biosimilar product is not required to have the same labeling as its reference product, and the biosimilar product may differ from the reference product for a variety of reasons. For example, the biosimilar product labeling should be specific to the conditions of use being sought for the biosimilar product. Therefore, the relevant data and information from the reference product that should be incorporated into the biosimilar product should be specific to the indications that are being sought for the biosimilar product. Furthermore, the labeling for the biosimilar product may incorporate some specific modifications to address the biosimilar product, such as preparation or storage, and information that does not otherwise preclude a demonstration of biosimilarity. It is also important to note that biosimilar product labeling must conform to the content and format requirements of PLR and PLLR, even if the reference product does not need to conform to these requirements. So generally, the FDA recommends that the biosimilar product labeling incorporates relevant data and information from the reference product labeling to the biosimilar product labeling with appropriate modification as discussed in the previous slide. Now, for studies conducted to demonstrate biosimilarity, FDA generally does not recommend that the biosimilar product labeling include that information. However, if a consumer or provider is interested in that information, the information is available on drugs at FDA under the FDA Reviews tab. And finally, FDA recommends that the highlights of prescribing information include a biosimilarity statement, and this statement is to describe the relationship between the biosimilar product and the reference product. And let's take a look at how this may appear in the label. This is a mocked-up label, just really with a skeleton of the highlights of prescribing information. So underneath initial US approval, there should be a statement that the product is biosimilar to the reference product. And please also note, even though you can't see it, but there is an asterisk after the word biosimilar. And the footnote would appear at the end of the highlight section underneath the last section, but above the revision date. Now, I'm going to very briefly touch on this topic, non-proprietary naming of biological products. There are specific guidances that speak to this topic. There's the original guidance as well as the updated guidance. So for specific details about how to approach this, please review those particular guidances. But generally, for 351A and 351K applications that are newly licensed, non-proprietary name consists of a core name attached to a suffix with a hyphen. And as communicated in the updated guidance for applications already licensed under 351A without a suffix and for transition products, FDA does not intend to apply the naming convention. So generally, the non-proprietary name of biological products include a distinguishing suffix to identify that specific product. Now, we're going to spend some time looking at product identification and biosimilar product labeling. This is important because we need to make sure that we're conveying information accurately. And as you'll come to see, product identification is very specific to the text being presented. So we're going to discuss the 3 approaches that are recommended in the guidance. And we're going to use a fictitious core name, replicamab, as the example. So FDA recommends the use of the biosimilar product name and the biosimilar product labeling for information specific to the biosimilar product and for directive statements and recommendations for monitoring, managing, or mitigating risk. If the biosimilar product's proprietary name is available, then you would use the proprietary name in those instances. And in example, it would be Neximio. If the proprietary name is not available at that time, or if you're referring to the drug substance, then you would use the non-proprietary name. For example, replicamab -CZNM. We recommend the use of the reference product non-proprietary name when you're discussing or describing clinical studies and data derived from the studies with a reference product. And in the example, it would be replicamab -HJXF. In instances when you're describing a risk or adverse reaction that is applicable to both the biosimilar product as well as a reference product, we recommend the core name plus products approach. And we recommend this approach even if that particular risk has not been reported for the biosimilar product at the time of licensure. And using the fictitious core name, it would be replicamab products. So this is a very busy slide, so I thought it would be helpful to look at an example to see how this may appear or how to do this in practice. So we're going to look at this fictitious example, and on the left column, you have the reference product labeling, and it's just 3 sentences. So it says, Junexant can cause acute hepatic failure. In clinical trials with Junexant, 10% of patients develop elevated ALT, and 5% progress to acute hepatic failure. Evaluate ALT at baseline and monthly during treatment with Junexant. So this is how the labeling would appear in a fictitious reference product labeling. So the question to you is, how would this information appear in the biosimilar product labeling? So you'll have to use the information that I just presented in the previous slide, and we're going to walk this sentence by sentence. So in the first sentence, Junexant can cause acute hepatic failure. We're discussing a risk that is applicable to both the biosimilar product and the reference product. So the approach that we will recommend is core name plus product. Okay, I'll give you all the answers right now. So in the second sentence, you're discussing the clinical trials with a reference product labeling and the data from that particular trial. So therefore, we will recommend the use of the reference product's non-proprietary name, replicamab-HJXF. And the last sentence, it is you're telling the provider that they should evaluate ALT at baseline and monthly during treatment. And so that's a directive statement recommendation for monitoring the patient for that particular risk. So the recommendation would be using the biosimilar product's name, and since the proprietary name is available, we could use the proprietary name. So this is how the label would appear on the right-hand side for the biosimilar product. So walking through this exercise, you can see that within one paragraph, or perhaps even within one sentence, you'll have to make the right determination of the approach to take based on the text being presented. And before I wrap my section up, I thought it would be helpful to give you some housekeeping tips. I'm always good about that. So in terms of how to, when you're responding to agency comments, and once again, this slide is more specific to labeling that you will receive via track changes, for example, prescribing information and patient labeling. So once again, follow the specific directions provided in the agency cover letter or letter. If it differs from what I'm saying here, follow the agency's letter, okay? But these are some general considerations and asks from us. So once you receive the agency's comments, please carefully review FDA's comments, edits, and if you agree, go ahead and accept those changes, including formatting and editorial changes. Now, this is the most important point. If you disagree with an edit from the FDA, do not use the reject button. Okay? Instead, keep FDA's comments, but instead you can propose a modified language or modified approach via track changes. And the important thing is please use the comments feature to describe why you're proposing something that is different so we can have a fruitful discussion. And as you're going through the labeling, there may be additional changes that you believe are warranted. Please go ahead and make those changes via track changes, and please be sure to include a comment. The last we want to do is kind of try to figure out why you propose something if there's no comment attached to it. And as you go through this exercise, please edit the format as necessary so the labeling looks very good and is compliant with the format requirements. I'm going to pass this off to Jess for the discussion on transition products.
So our last substantive topic is going to be labeling and transition products. So as I promised at the beginning, and I'm sure you've all been waiting with bated breath, just some additional background about transition products. Certain proteins have been approved as drugs under Section 505 of the FDA Act. The most commonly known ones are insulin and insulin analogs and somatropin, but there are also a number of others. As explained earlier, The BPCIA Act amended the definition of biological product in Section 351 to include a protein except any chemically synthesized polypeptide. In addition, the BPCIA Act also provided that an approved application for a biological product under Section 505 of the FD&C Act shall be deemed to be a license for that biological product under Section 351 of the Public Health Service Act as of March 23, 2020. FDA has issued guidance that's explained that approved applications under Section 505 for deemed products will be transitioned to 351 BLAs. So in short, overall, any biological product as defined in Section 351I of the amended Section 351I of the Public Health Service Act that was approved in a Section 505 application under the FD&C Act will be deemed to be licensed under Section 351A on March 23, 2020. Additionally, applicants can also seek licensure of under Section 351K of products that are biosimilar to or interchangeable with a transition reference product. In December 2018, FDA issued draft guidance, the deemed to be licensed provision of the BPCIA Act, questions and answers. And in that draft guidance, there was some discussion about transition products and labeling. And most labeling requirements are the same for BLAs and NDAs. There are some minor differences, and the guidance, or the draft guidance, has more detail if you would like to have more additional information about that. One example is that both the container and carton labeling, as well as the USPI, must include the proper name of the biological product and the license number. And note that FDA will designate the proper name of the biological product in the license. And as noted in an earlier slide that Ruby discussed, as discussed in draft guidance, FDA does not intend to apply the naming conventions to the transition products. So in other words, the proper name that was designated in the license would not receive the distinguishing four-letter suffix. In addition, the guidance announced a compliance policy for the labeling of biological products that are the subject of deemed BLAs. The goal of this compliance policy is to help minimize any possible disruption and burden associated with reconciling those minor differences in the NDA and the BLA labeling. Overall, FDA does not intend to enforce the BLA labeling requirements for transition products until March 23, 2025, so it's 5 years after the transition date. One thing to note, though, is that if during the compliance period, a deemed BLA holder submits a supplement with proposed labeling revisions, the BLA-specific labeling requirements will need to be addressed before that supplement can be approved. Additionally, to ensure that there's sufficient time to comply with the BLA-specific labeling requirements, FDA recommends that submission of a prior approval supplement be done within 2 years after the transition date. And also to help ease burden, FDA recommends coordinating any changes to the container carton labeling with the changes to the USPI. So we're going to close with a challenge question. And you're going to see some familiar names that I'll be using the same fictitious product that Ruby mentioned before. We're not that original. So here it says the labeling for Junextant, replicamab, -HJFX states, treatment with Junextant increases the risk of serious infections involving various organ systems and sites that may lead to hospitalization and death. And the question is, which product identification term would be consistent with the biosimilar labeling guidance for the labeling of Neximio, replicamab-CZNM, a proposed biosimilar to Junextant? And since I'm sure you all are paying very close attention, if anyone wants to shout out what A, B, C, or D, any brave soul? You guys are really on your game this morning. That is correct. Because this is giving the information as a benefit risk profile that's applicable to the biosimilar. D would be the correct answer, even if that adverse event had not yet occurred with the biosimilar when the time of licensure. And with that, we can now open up for questions for you guys.
All right. People that are new online, hopefully you've been typing in your questions to the Q&A pod down in the lower right-hand corner of the screen. If not, now is the time to do so. Please take the time to type in nice and slowly so that it's easier for our people reviewing those questions to read. Everybody in the room heard the spiel before, but just as a reminder, what we're looking for are questions that will be of value to everyone based on the topics that were just covered by our presenters. So please no very specific to your application type questions. But don't let me scare you off like I did yesterday. If you have questions, please come down to the microphones. Let's go ahead. Do we have somebody making it? There we go. All right, we'll start over here on microphone number 2, please.
So in the example on slide 14, when you compared how the adverse events would be shown for the innovator's biological product and then how it would be shown for the biosimilar, it looks curious to me because I know working in drug labeling, when you're describing adverse events or warnings or even non-clinical data, and sometimes when you use the non-proprietary name, FDA comments back that you should use the trade name consistently throughout the labeling, because it would be confusing to readers or prescribers, or that you may be discounting or undervaluing risk information in the label. Is that a concern with not using the biosimilars name consistently as shown? Because it seems very different than the feedback we receive for drug labeling.
Oh, so you're referring to the second sentence?
Yeah.
And in the second sentence, you're actually referencing the reference product. You're using the non-proprietary name of the reference product.
Right, but you're using replicamab twice and then only using Niximio at the end. Where I feel if I did that in drug labeling, it would be common for FDA to say that I should be using Niximio throughout.
I think for,
Yeah, so I think the difference is in the second sentence, you're specifically discussing something that was, you're discussing clinical trial with a with a reference product labeling. And in this instance, you're discussing the clinical trials from the reference product, so then you would want to use the name of the reference product, and we're just happen to use the non-proprietary name of the reference product, versus the third sentence, you're discussing just some recommendations about how to monitor for that risk, so then you would use the biosimilar product's name. Does that make sense? So I don't know if it was confusing because the second sentence, the red is actually the reference product's non-proprietary name.
just to give additional thought processing, biosimilars are different than a 351A drug or a 505B NDA, and between that and the naming conventions, there are different considerations that are going to govern the labeling and how we communicate that information. But we appreciate, and it's helpful to have the feedback to make sure that the information that's getting out there is going to be helpful, informative to both the prescribers and the people receiving the medication.
All right, microphone number 2, nice and close for me, please.
Yes, for a biologic that requires special handling, such as a cryopreserved product, where is the best place to include special handling instructions for the user, which in this case would be a clinic or physician? It's not something that the patient would be bringing home with them.
Yeah, I would think, once again, a lot of things are fact-specific, but usually you will see some special handling. I would think in Section 3 and 16, we're talking about the specific use of the drug product, but that's something you would discuss with the review division at the time of the review.
All right, thank you very much.
All right, we've got about 3 minutes. If there's any other questions, come on up to the microphone. Otherwise, let's go to our first online question, please.
You stated that generally, biosimilar product labeling should not include a description of data from clinical studies conducted to support a demonstration of biosimilarity. Why is that?
That's a good question. So I think perhaps we can go back to how Eric Brodsky was discussing prescribing information yesterday. And the intent of the prescribing information is to communicate to the healthcare provider and the practitioner about how to use the product safely and effectively for the indicated indications in the label. And so if you take a look at the biosimilar development program, the clinical studies conducted for the demonstration of biosimilarity is not designed to independently evaluate the safety and efficacy of the drug product. You're looking at perhaps different patient population, different endpoint. And if you include all that information in the label and a provider is not familiar with the demonstration of biosimilarity, it could be confusing. And in addition, the data may be misinterpreted. So the recommendation is that you include the information for the reference product, and if someone is interested in data used to support biosimilarity, the information is available on drugs at FDA.
Okay, do we have another online question?
Yes. At what point during the labeling review process would a sponsor receive the 4-character suffix so we may add it to the labeling, container labels, and carton labeling prior to PDUFA?
Yeah, there is, I have DMEPA folks here. So there is a process for that. And usually they're looking at the proprietary name, they're looking at the suffix. So you definitely will receive that information prior, before the go date. So there should be sufficient time for that to be vetted.
Okay, microphone number 2, please.
Yeah, my question was, how is that 4-letter suffix generated? Is it like a random number generator, or how is it put together?
Mm-hmm. Okay, so there is the guidance on non-proprietary name is very detailed. Even with the previous question, it talks about how you would go about doing that. So you can definitely propose, or the agency can come up with that determination. So there are different approaches to the suffix, and then there are criteria about what it has to be meaningless, things like that. So
Yeah, that guidance will have a lot more detailed information about how to go about that.
All right, we've got time for one last question, please.
Hi, good morning. This is Danielle Harris from the Division of Medication Error Prevention and Analysis from DMEPA. So I just wanted to address the last question. First of all, in terms of the timing of the suffix, we are targeting to try and get suffixes reviewed or designated by mid-cycle or shortly thereafter with regard to the biologic review timelines. And with regard to the question that you just answered, not much to add. We do use that guidance if we're designating. There's criteria laid out in the non-proprietary naming guidance that explains those suffixes. It's not necessarily a random generator. There are specific criteria, and then we apply those criteria to any suffixes that are submitted for our review.
thank you for the lifeline.
Thank you so much for that clarification, and please help me thank our 2 presenters for their